Good morning, viewers at home. My name is Olatunde Michael. I'm the team leader of Omega BioEdutainment. This is another educational series in in Omega BioEdutainment. Today, we are going to continue our discussion on general microbiology. The last time we met, I was able to really tell you a lot of things. I think we started with the last thing we discussed was to look at what are the basic laboratory techniques in so um, on general microbiology. The last time we met, I was able to really this afternoon we are going to actually talk about a general review, general recap of where we stopped last week, and today's topic is going to be on microbes, that is microorganisms and diseases. That is, what are the roles that microorganisms play in causing infection. So we are going to see that today. Yes, well, before we go on, we need to take note of the following: that definition. Okay, there are certain terms and terminologies that we have to get used to. At this level, whenever you come across microbes, okay, when we are talking about the history of microorganisms, you will recall that I made mention of some early scientists, and these early scientists were part of they arrived at referring to microorganisms as microbes. So don't forget that according to the early scientists who called microorganisms as animalcules, he called them animalcules. That is, they believed then that microorganisms could be grouped into the group of animals, and that cools, that is, they are in form of particles. So before they are able to now continue and know a lot of things about it. So today, microbes, we are still referring to this. So organism, that is microorganism that causes disease causing microbes are referred to as a pathogen. So patho is coming from pathology, which has to do with diseases. So disease causing organisms are called pathogens. Not all, when you look at microorganisms, microbes, okay, you can divide them into two. That is non-pathogenic, and those ones that are also pathogenic. So that means more non-pathogenics are those organisms or microorganisms that can perform or serve as a other beneficial, that are beneficial to our health, whereas those ones that are in this group known as pathogens, that is those ones that cause infection, they are called pathogenic. Now the now the first thing I told you is what are microbes. I've told you. Then what are pathogens? That's number two. The third one is you will also come across certain things like what do we call the ability of microorganisms to cause diseases. Ability of microbes to cause diseases at this level are called pathogenicity. Okay, so that is the ability of an organism to be able to cause infection, and we can give examples in each case. Non-pathogenic are majorly members of lactobacillus, because members of this group are those ones that we normally use in our fermentation, that is to ferment our food, as well as this. So lactobacillus species. Then pathogenic ones are include salmonella. We have a lot of them. Salmonella. You have mycobacterium tuberculosis, the one that causes TB. Okay, so that is that. Now the next thing is what actually determine whether a microorganism is going to be pathogenic or not. You see, what we are saying in essence is that it's not all, depending on the habitat, depending on the area that they find themselves. So that would now depend solely whether they are going to be disease causing organism, that is whether they are going to be pathogenic or they are not going to be pathogenic. So we need to let that one get sink into us. So those ones that are non-pathogenic, this one, apart from some of them being used in fermentation, some of them are used as nutrient cyclers. They help to recycle some nutrients. So those ones are part of so if they are found in the soil, so that means those ones that are going to be nutrient cyclers must be found in the soil. They must have their role that they will perform in the soil. So please, we need to take note of that. So now I have mentioned what microbes are, that is microorganisms. Another word for microorganism is called what? Microbes. So those ones that causes diseases are referred to as pathogenic, and those ones that are not pathogenic, that means they are beneficial. So that is why I said you can. So if you have a question whether microorganisms, microbes, are they our friends or foes? Okay, are they friends or foes? If they are our friends, that means they are non-pathogenic, that they are beneficial, and we have a lot of them. Those ones that help us to recycle our nutrients, those ones that help us to digest some vitamins that naturally or normally we cannot digest. So all these ones are non-pathogenic, and those ones that can cause or initiate diseases are called what? Pathogenic. Then the ability of any microorganism to cause disease is called pathogenicity. So we need to know that. Another thing that I want you to know is virulence. If they say one microorganism is more virulent than another, what are they saying? That is, Virulence refer to the degree or the gravity. Okay, that's the word. The gravity of causing infection. The gravity of causing infection is often referred to as their virulence. So you can easily say that maybe Mycobacterium tuberculosis is more virulent than bacteria. And I have told us one of the basic factors that determines whether microorganism is going to be beneficial or is not going to be beneficial. That is the environment they find themselves. Okay, so I want us to really understand and know that. So having solved all these puzzles, that is most of the definition we have, terms and terminology that we have been able to see. Okay, we will now go back to how does or how do microorganisms, how do they cause infection? How is it that they have to make use of punches? Is it that they have some needles that they will just inject into our body? How do they cause this infection? So that one is going to lead us to another terms that we are supposed to know. That is toxin. Toxin. So toxin are substances. Toxin are substances that microbes microbes release into the environment. Don't forget that the environment can also be inside us, can be outside us. So the toxin, their weapon, okay, that's another. Their own weapon is referred to as a toxin, weapon of mass destruction. Is a So toxin are substances that microorganisms release into the environment to cause infection. The weapon, their own weapon, is called toxin. Okay, so from there we will now move on to another thing. There are two basic types of toxins. Two basic types of toxin that we know. Toxin, there are basically two types. It can be classified into two. We have endotoxin and the exotoxin. Okay, endo and the exotoxin. From the word endo, that is internal. That means when you have, okay, internal means that when you have a microorganism like this, let's say you have a microorganism like that. Now, if the release of that toxin is now towards, if they now release toxin, if the release of that toxin is internal, that's why I'm using red pen so that you see that is a toxin. So that is a endotoxin. Endo, within. So that is that. Now when you now have exotoxin, is when you have a bacteria. If you let's say you have a bacterial cell like this. So if if what you now have is that there is release of their weapon into the outside, that is exotoxin. That is exotoxin. So that is that. And basically, no, although not at your own level here, basically endotoxin is basically found in gram negative, whereas exo is found in the among gram positive. Please, at this point, just take note of that. Okay. Now, this is how microorganisms, how microbes cause infection. So they have two weapons. Their weapon is known as a toxin, and this type of toxin are basically of two types. Don't forget that at this level, I told you that you should just rely on the elementary classification or the primary classification of microorganism into gram negative and gram positive. I told you in the last lecture that whether they are going to be gram positive or gram negative will depend on their cell wall. Those ones that have very thick cell wall, they are called gram positive, and those ones with a thin cell wall, thinner, called gram negative. So as I said, just like that, we now have endotoxin and exotoxin. Depending on the site or the location, depending on the targets of the toxin. If the toxin is targeting its release to still be within the bacterial cell, that is called endotoxin. But if the target is towards the environment, the release of his biological weapon into the environment, that is called exotoxin. And I said exotoxin is primarily found in gram positive, whereas endo is primarily found in the gram negative. Now, that is how. The next question that we need to ask ourselves is that what are the roots, not source. So roots, look at the word roots, the roots of infection. Now we can call roots the same thing as parts. For example, now if you want to go to Lagos, what route will you follow? If there is going to be an infection, what are those routes? What are the parts, the roads, the junction that this infection process is going to pass through? Now, don't forget that I told us something about pathogenicity. I said that pathogenicity is the degree, is the degree or the ability of a given microorganism to be able to initiate, cause an infection. And one thing is very, very important, that the first thing is, that means microorganism should be able to invade, to have the ability to invade, to enter. For example, now, for any successful robbery to take place, that means the robbers must be able to enter. That is invasion in disease process. Okay, so the first root of infection is that the first stage of infection is adherence. That is ability for microorganism, for bacteria, for viruses, for any microorganism to be able to stick, attach, to be able to attach to the surface. So that is adherence, that is the ability of the first thing that will happen is that microorganism should have the ability to stick, to come in contact. If there is no contact, there cannot be any infection. If there is no adherence, if the microorganism do not have anything to stick on, just like when you want to fire a gun and you don't have any solid ground, all these powerful rifle, you want to fire powerful rifle, there are some that even before you shoot, you have to lean on the support. So you have to stand very well. You have to hold your strength. So the same thing with infection process. So the first one is adherence. Okay, and what they normally use for adherence is they use fimbriae, flagella. You can use flagella. Then apart from using flagella, so this one will help them to be able to stick. They will be able to stick, just like bacteria that causes a plague, that is tooth decay. So there are some that the first thing is that their own weapon, their own toxin is to be able to secrete sticky substance that will help them to be able to attach firmly, so that come any condition, they will not be swept away. So that is adherence. So the second one is colonization. That means as soon as they get themselves to attach themselves to the surface of their host, the next thing is to multiply. The next thing is to increase their own number, and they will increase their own number to the level that they will now be the one that will colonize it. They will be the one that will take the territory. They will take the control. So that is what we know we call colonization. So ability of any microorganism to be able to multiply and be able to dominate in any environment is called colonization. After that, after colonization, the next thing is they should have effective mechanism, efficient of host defenses. So the next thing is that they will be looking for a way. Don't forget that once there is any infection, or once the body senses strangers within the whole cell, the next thing is that your system, the human immune system, is going to be triggered. And whenever it is now being triggered, the ability of a microorganism is to be able to make sure that whatever the host might do, the immune system will not be able to catch up with them. So one of the main objectives of an armed robber is to make sure that they are not being caught either by night guard or by whatever. So that is what is called efficient. Okay, so and their success in efficient would actually depend on the depends on the state of immunological state of the host. If the host is immunologically competent, that means colonization will not take place. But if by one reason or another the host immune system is already reduced, is already lowered, so what is going to happen is that colonization will take place, and once colonization has taken place, they will now take control over it. Now I want to Now start giving examples. Don't forget, the first thing we did was to look at certain terms and terminologies in diseases, microorganisms that cause diseases. So we are able to look at them. What are microbes? What do we understand by pathogenicity and non-pathogenicity? What do we understand by then examples in each case? So the next thing is now we want to look at what are the examples or case studies on diseases and microorganisms. So the first one is don't forget that microorganisms are divided into four. The first one is infection that are caused by bacteria. They are called bacterial infection. An example is that of Mycobacterium tuberculosis, Streptococcus pyogenes, the one that causes sore throats. It's Streptococcus pyogenes. The next one is viral infection. The next type of infection is viral infection. And under viral infection, there are different types like coronavirus, like influenza, and so on and so forth. So that is that. Now, the next thing is a fungal infection that are caused by fungi. Okay, fungal infection. An example of that is athlete foot. That is a common infection that affects the feet, as well as making them to eat us. That is that. Then the fourth type of group of microorganisms that are pathogenic are parasitic infection, and we have prime in that is malaria parasites. Malaria is an example that is caused by Plasmodium species. That is that. Okay. Now, the next thing is how can we prevent diseases? How can we prevent diseases? The first one is PPE, personal protective equipment. When you are working with microorganism or you are working in the lab, so what are the protective mechanisms? How can you protect yourself so that you do not inhale or you run away from all these microorganisms that cause different types of diseases? Okay, so that is the first one. PPE is the first one. Then number two, the second aspect is containment. Contain. You make sure you in advanced laboratories there is something they call containment, whereby you will not have a direct contact. You will not be working directly with a microorganism. You'll be working there as well as so that means you are being contained in a place. So if your containment process is excellent, but your waste disposal is one thing. So waste disposal, proper waste disposal is another way by which you can also prevent yourself from getting infected. Then the last one is decontamination. There are some chemicals from the laboratory that you will not just go and trash them into the dustbin directly. No, you have to decontaminate them. Example is sputum that they normally use for to screen for TB. you have to decontaminate it so that you make it less infective. You are making them to be less infective so that anybody that comes across them is not going to get himself or herself get himself infected. So that is very, very important. Okay, now um we so that we will not just be overburdened. So I want you to go and read up preservation techniques. Read preservation techniques, okay? And I'm going to give you assignments on preservation techniques, okay? Um, I won't write the assignment. I will just dictate the assignment for you so that later you will go and do when you listen to the video, you'll be able to do your assignment and submit. You just have to submit in the comment section. Submit in the comment section. Okay. Now on preservation techniques, what do you understand? What do you understand by preservation techniques? Preservation techniques. Okay. Then what are the types? What are the types of materials that can be preserved? What are those materials that can be preserved? Then in the laboratory, they normally preserve cultures. Why do we have to preserve cultures? So please, you have to just work on these three. The first one is, what do you understand by preservation techniques? Okay, why are cultures, different cultures, why are they so important to be preserved? And differentiate between traditional method of preservation and modern method of preservation. So I think having said this, we want to really come to the close of our contact hours or contact time on general microbiology. So if any one of you have if you have any question, I think you can direct that question to me in the comment section, and I'm going to appropriately respond to that. However, the assignment given to you should be submitted not later than 10:30 a.m. on Monday. So that means you have been given an ample time of more than 48 hours. And 48 hours, so that we'll be able to provide that assignment. Okay. So let me also mention that the last lecture we stopped at selective media. So the next one should be differential. But just like I told you that I don't want to overburden you with too many or too long notes. That is why it is at this point that I want to really stop, and so that I will be able to. So don't forget to subscribe, like, and share this.